Welcome everybody. My name is Andrea Decker. I am the Adult Programs Manager at the Fleet Science Center, and I'm super excited that you're joining us tonight for another installment of our special Suds Science series on the race of San Diego to help find a cure to COVID. I'm super excited about this series for many reasons. First of all, I'm glad we can bring science to you online. While I would love to see everybody in real life, I'm glad that we can at least still offer science engagement online. The other reason that I'm really excited about this series is because it helps us shine a spotlight on San Diego science. We have a tremendous science resource in San Diego, and I'm not sure that all San Diegans know about it. And as unfortunate as this crisis is, and it really, really is, it's great to have such a science resource in San Diego that is working to help us find a way through the crisis. So today is one of the, actually now 4-part event. It was supposed to be a 3-part event, but we added one more event at the end of June, so I'm very excited about that. But I'm glad we're having the second event tonight. For the first one, we looked at a company here in San Diego that's working on a vaccine. For the second one, we're looking at treatment. Just as important as finding a vaccine is being able to find a treatment that will help people who have caught the disease and are sick with the disease now. It's a very severe disease in many cases. So I'm glad that we have researchers with us today from the Sanford, Burnham, Prebys Medical Discovery Institute. And I'm going to just introduce them by name, and then they will take it away and explain a little bit more about the work they do. They work with lung organoids to look at how the disease works, and then also have researchers who look at a vast catalog of drugs that are already available and FDA approved that might work for this disease that we could quickly repurpose and use to help people with the COVID disease. So without further ado, I'm really excited to introduce the team that we have with us tonight. I'm going to start with just how my screen is laid out. So I have Dr. Sandra Leibel with us today. Welcome, Sandra. Thank you so much for joining us. On her team also is Dr. Snyder. Evan, thank you so much for joining us tonight. Also with us is Dr. Riva. Hello, Laura. Thank you so much. And we also have Dr. Xin Yin. Hi, Xin. How are you?
Good. Yeah.
So the way that tonight will work is Sandra will start with a presentation of her work that will then nicely lead over to what Laura does in her group on finding, on looking at the catalog of drugs. The presentations will go until about 7:40, 7:45-ish, and then that leaves us with at least 15 minutes for Q&A. If you have a question, the chat function is disabled, but we have a Q&A function that you can use. If you have a question, you don't have to wait until the end. You can just type it into the Q&A section. There's little 2 speech bubbles on the bottom of your screen. Just open that window up, type in your questions, and we'll get to it in the Q&A at the end of the presentation. But so you don't forget your question, type it in as soon as you have it, and this way we'll go over once we have it. Wonderful. And with that, without further ado, I'm going to turn it over to Sandra.
Thank you very much. Hi, everyone. Hello, virtually. I am Sandra Leibel. I'm a clinician scientist. Clinically, I work as a neonatologist taking care of preterm babies and term babies, along with Evan as well, who's also a neonatologist. As a scientist, I'm a lung biologist, and I actually work in Evan's lab, who's the director of the stem cell core. And we're interested in induced pluripotent stem cells, and I'll describe what those are in my talk, and how you can make different organ systems to study development as well as disease. And my specific focus is stem cell derived lungs. So I'll start sharing the presentation. Okay, so as you can see in this picture, these are what my little lungs in a dish look like. If you want to take a look at what we see through the microscope when they're developing from the stem cells. And initially I was using these to study a disease that happens in preterm as well as term babies regarding a specific soap that we make in our lungs called surfactant. And this soap is what allows us that when we breathe out to keep our lungs maintained in the open state. So every time we breathe out, our lungs don't collapse. And that's because we have this specific molecule called surfactant. And preterm babies lack that molecule. And so they're at higher risk of lung damage. And so I was studying that in a dish. But what's also from the standpoint of COVID-19 and the virus SARS-CoV-2 is that it attacks the cells in the lung that actually make surfactant. And people that get really sick and need to go to the hospital and end up on ventilators, they have this disease called acute respiratory distress syndrome, or ARDS. And that also hits the surfactant system and makes it really difficult for them to be able to breathe and keep their lungs open, which is why some of the time they do have to end up on ventilators. So we thought this would be a great model to study not only how this disease occurs after infection, but also if we can develop a therapy in collaboration with Dr. Chanda's lab and with Dr. Xin and Riva, then this would be great to actually model in a human lung instead of the various animal models that we usually model our medications on. So first things first, don't worry about all the words. I just found that this is a beautiful diagram that actually explains everything that occurs during the infection. So I just want you to focus more on the picture itself and not necessarily all of the different specifics in these little words. So what this top picture shows is what a healthy alveolus looks like. So the alveolus are little grape-like sacs, and that holds the alveolar cells. And what the alveolar cells do is there's these really flat ones called type 1 cells, and they're the ones that function in gas exchange. So they collect the carbon dioxide from the capillaries or the blood vessels that the cells are bringing back from the body, and then this carbon dioxide we blow out. And then what they also do is they allow the oxygen that we breathe in to cross this membrane back into the circulation, and that goes back to our cells to feed them with oxygen. We also have type 2 alveolar cells, and these are the kind of more heftier blue whitey ones, and these are the cells that actually make surfactant. So these are extremely important, but both, as you know, are very important. So this alveolus is usually just functioning by exchanging gas and making surfactant to keep the lungs open. So what happens when the virus SARS-CoV-2 comes in is it has these specific receptors on its membrane, and these proteins actually detect specific receptors on the type 2 cells specifically. And so they'll latch on to the type 2 cell, allow entrance, and then that's when they actually start causing damage. So there's different levels of damage to the lungs, and that's why we see different clinical symptoms in some patients. Some patients don't show any signs at all, which means that it probably didn't infect their alveoli. They probably maybe just entered through their nose and didn't actually cause any damage. There's other people that get infected in the alveoli, but their immune system can handle it and probably beat it off. And then there's others that actually have more severe disease. And this is what happens in the more severe disease, is that because of all the damage that it wreaks, the immune system is called in. So these are macrophages that are coming in through the blood vessels into the alveolus in order to help with this infection. And the combination of the damage that the viral causes, causing this kind of leakiness in that membrane that needs to be nice and intact, causes fluid to start to accumulate within the alveolus. And then if a person has an immune system that is maybe a little hyper-reactive, then the immune system, kind of by way of accident, tries to secrete the proteins that it does in order to kill the virus, but then it also compounds the damage that occurs within the alveolus. And so not only does this fluid and the damage occur, but you can see that there's also a thickening between the alveolus and the actual capillaries here, the vasculature, and that causes decreased ability to actually exchange oxygen and carbon dioxide. And that's why a lot of these patients require supplemental oxygen, whether through a ventilator or just a high flow nasal cannula. And then finally, when we get severe disease, you can see that this alveolus is completely sick. It's not nicely expanded anymore. It's pretty much not useful for any of its functions, such as gas exchange. And so this, what we see when we look under high intensity microscopes, is that it's just filled with just protein gunk, lots of immune cells that were coming in trying to save it, but ended up being unable to. And so this alveolus functionally is completely hurt and injured. And so the way that we usually model disease is we can't just, you know, take a little piece of somebody's lung, because the lungs are obviously behind the rib cage. It's very difficult to do a biopsy. In skin, we can easily biopsy skin and grow it out and study it. Hair, you know, anything outside the body is very easy to obtain and study in a human person, but anything more internal like the heart, lung, liver, pancreas is a lot more difficult to study. And that's why we've always used either mouse models or other animal models similar to us. There's even some labs that use zebrafish or yeast, any type of organism that does something similar to how we do it. They're fantastic to study because there's a lot, they breed with more pups versus you know, having human tissue and trying to grow, and some human tissue can only grow for 2 weeks, and then, you know, it just changes too much, so we can't actually model what we want to model. So they're just more easier to handle and study. But the mouse is not a human. And that's why when this a new way of reprogramming what's called somatic cells. So somatic cells are anything that's already been differentiated fully, like your skin. We can also do this in white blood cells from your blood. We can actually isolate these from a person, and then we can add specific reprogramming factors inside a dish and reprogram fibroblast or skin cells, as well as white blood cells, into these fully functional, what's called induced pluripotent stem cells. And these are very similar to embryonic stem cells, which is how we all started from. And embryonic stem cells give rise to every single tissue, organ, cell in our body. And so now we can make these without actually having to go and obtain embryonic tissue. And so we can take your skin cells, make iPS cells or induced pluripotent stem cells, and then actually differentiate them into any type of organ system that we like to. So we can make blood cells, we can make cells that are in the lungs, such as sclera, as well as alveolar type 2 cells. These are the ones that secrete surfactant. And then what we're trying to envision for the future is either transplantation. So if we give back your own cells, your body's not going to reject them. So it's very ideal from the transplantation standpoint. The only problem right now is that everything's super miniature mini. So, you know, my lung little organoids, they're pretty much the size of the tip of my pinky. So giving that back to me is not going to have any impact at all. So we're still far away from transplantation, but what we're not far away from, and what we're actually actively pursuing right now, is this high throughput drug screening. So we can put these little organoids in a huge plate with lots of different wells, put them through a big machine and test a lot of chemical compounds, as well as adding the virus, and actually just like looking to see whether the virus infects, whether it does not infect, and which chemical compound was placed at the same time. And then that way we can do this high throughput screen to look for therapies. So this is just a quick view of how we make these stem cell derived organoids. So it's a process that takes over a month. We start with the stem cell, and then we just follow lung development like it happened in all of us when we were a fetus and an embryo. And these are just pictures. So once they get into 3D, then they start to grow out like little tiny lung buds. You can see they start branching. So we call this organoid induction. This is branching, like the lung goes through. And then we add a maturation agent, which is a steroid. And we actually give this to moms who are coming in preterm labor. And we actually give the mom steroids in order to mature the lungs of the fetus inside of her. So this is something that we do clinically, and it also works in a dish as well. So we give the little lungs some steroids, and then you can see that they start to change, they start to mature, and they start to alveolarize. So they start to make mature alveoli. What we can also do in our model is not just have the lungs on their own, but we can co-culture them with cells that make blood vessels. And this is a picture of the blood vessels in green. And then we can also co-culture them with various immune cells. So this is a picture of the lung organoids co-cultured with macrophages, which are specific immune cells resident to the lung. And so this is just a video of that maturation phase of the lung organoids going from a branching morphogenesis into an actual mature alveolarization. And this is three-dimensional organoids. So you'll see the different fuzzy ones are at just a lower level of the matrix that they're in. So That's that video. And then we also, some of our organoids were actually able to what we call breathing. So I don't know, it's pretty subtle, but if you look at this big guy over here, it's kind of breathing in and out along with the other one right beside him over there. So it does mimic the lung in a dish, albeit extremely tiny. And so what is so exciting about using these lung organoids is not only to fight this disease and to understand what are the cells that it infects, what does it do inside the cell after it infects? Can we make therapies targeted to, number one, preventing it from entering the cell, so kind of a prophylactic approach, or can we actually see what it does to the lung cells versus the immune cells, and then have therapies for multiple points in the disease process? So, you know, usually all the clinical trials right now are using monotherapy or one drug for pretty much everyone. It doesn't matter if you have that moderate disease, the severe disease, or the pretty deadly disease, they're putting you all on the same drug. But with our lung organoids, we can actually determine that if you're at the earlier stage of the disease, maybe this drug will be better versus if you're more to the end or the more severe aspects of the disease, then maybe we could try a cocktail of these different medications that target both immune cells as well as the respiratory or the pulmonary cells. So that's what's really also exciting about using this to study this disease. What's also really interesting is that we're all different people. We have different genetics, we have different dispositions, we've had different lives. Some of us have smoked, some of us have not smoked, some of us have diabetes, some of us don't. And so again, this one kind of shotgun approach may not specifically work depending on your gender, your ethnicity, and what we call your comorbidities or the other illnesses that may be going on in your body. And so if we can actually take specific cell lines from men, women, young patients, old patients, all the different ethnicities that show different ranges of how they're tolerating or not tolerating this viral infection, then we can actually target these therapies or prophylactics for the actual patient-specific population. So that's another fantastic use for iPSC-derived lung organoids. It's also, because it mimics a human organ in a dish, we can just keep making them. So I also grow primary human organoids. So I take human tissue from biopsies. I can grow them as well, but I have to be, you know, I wait once a month for a biopsy to happen. Sometimes they give me good tissue, sometimes it's not very good because maybe the patient had cancer. And so it's good that they're taking it out, but then I can't actually use it to grow. But if I use these iPSC-derived lung organoids, I'm making these. We start these differentiations every week. So we have an incubator full of these little lung organoids that we could study, add things, take things away, manipulate them. You can also study genetic defects. So there's actually a disease that's affecting the pediatric population or kids where they're showing that inflammation is hitting their hearts in a specific way that we see in pediatrics in a disease called Kawasaki disease. And they're wondering whether this is due to a specific gene mutation. So what we can do is we can take our induced pluripotent cells, knock out that gene, and then make the different cell types that are affected by this disease and see whether it is truly that gene that predisposes these kids to this disease effects. So that's something else that we can do within this lung organoid platform. Finally, like I already said, some people have smoked their whole lives, so they have very different lungs. So we can actually do that in collaboration with another laboratory at Sanford, Burnham, Prebys. They actually have a machine where they add vaping chemicals. And then we could put our lung organoids into that machine, expose them to those chemicals, and then study the effects, and then study how the virus infecting lungs that are exposed to these chemicals react to the virus. And then I already mentioned drug screening. So we used to do it in animal models, but a lot of animal models, they actually show positive results, and then we translate them into large clinical human trials, and we get nothing. They don't show the same results as we saw in the mouse. So it's definitely frustrating, but it's all we have as scientists. But now if we can actually have a true human model of the disease that we're studying, then it's actually a lot more believable, or we have a lot more hope that this truly will work. So it's nice to avoid that animal model and go straight into determining whether the different medications will be effective in a human model. Finally, It's great working at SBP because there's so many collaborations going on. Everyone's studying different parts of the human body. And we've all come together during this crisis to really put our brains together and to work together as a community in order to find a treatment, so then we can actually beat this thing. And so you'll get the presentation from Dr. Riva about the high throughput drug panels, but we can also, as I said before, we can make different organ systems as well. So this virus not only affects the lung, but we've seen patients describe encephalopathies, which means an inflammation of the brain or the heart or their immune system, like the neutrophils. And we can use these induced pluripotent stem cells to make human-derived various other organ systems to study the effects that SARS-CoV-2 imparts on them. And so that's my presentation for what we're doing in modeling this disease.
Wonderful. Thank you so much, Sandra. And with that, Laura, I'm going to turn it over to you.
Okay, I'm going to share my screen. So I really enjoyed the presentation from Sandra. Mine will be a little bit different because, of course, we are doing different things. Is a different work, right? It's we are looking for antivirals able to block SARS-CoV-2, and of course, having people developing such a great tools like Sandra and Dr. Snyder are amazing tools that we can use to confirm what we are looking at the large scale in our case. So today I'm going to describe you a little bit the journey that brought us to run large-scale drug repositioning screens against SARS-CoV-2 in order to identify antivirals. So let's see. Okay. So I will start from the really basics of everything. So what happens when a person gets in contact with a virus? So either it fights the virus and it doesn't get infected, but what if the person gets infected, right? Well, it's great when there is a vaccine able to block the virus. So a vaccine stimulates the production of antibodies, and these antibodies will be able to recognize the virus once the person will get in contact with it. And these allows the person not to get sick. So ideally, a vaccine is always the best solution to fight a viral infection. But what happens if no vaccine is available, or if the vaccine is not accessible to the entire population, or if a virus is highly mutating, or if you're facing a new outbreak, which is exactly what is going on right now, right? So yeah, in the long term, the vaccine is the best solution, but we also need to find other ways to block viral infections. So what if then there is no vaccine? What is the other way to fight a viral infection, right? Well, we need to find a way to block a virus once already got inside people. And this is what is done actually by antivirals. And today I'm going to talk to you about antivirals. So antivirals, the definition of an antivirus is an agent that kills a virus or that suppresses its ability to replicate, and then as a consequence inhibits its capability to infect and reproduce. So antivirals can block a virus in different ways because a virus to survive needs a cell. So the way to block a virus is either to target the virus directly, so one of the protein of the virus or one of the processes like one of the enzymes of the virus or the RNA of the virus, or block something in the cell which is necessary for the virus to replicate. So antivirals can target different things, either cellular pathways and proteins or viral pathways and proteins required for the replication of the virus. So the drug discovery and development process is a long journey in general. So people start from chemistry, synthesize different molecules, and from there on, they start testing these molecules for the disease or the infection they want to look at. And this applies not just to viral infections, but also to each kind of disease in general, from cancer to viral infection to neuro. And so, as I was saying, it's a really long journey, starting from the synthesis of a molecule until the approval and the post-marketing surveillance of the drug. And this journey can take up to 10 to 15 years because the synthesis takes time, the optimizations for having a drug targeting a specific disease also requires a lot of time. And from then, once you have something working in a system that is called in general in vitro, so in the cells, in the labs, as we test in general, there are still a lot of steps that needs to be performed and went through before having a drug on the market, because it's necessary to prove that a drug is safe. The fact that a drug is working in a simplified model doesn't mean that it will translate it in an entire organism. So yeah, this takes a lot of years. But as you can all understand, in situation of outbreaks and dramatic cases like the one that we're facing right now, we don't have 10 to 15 years to find an antiviral able to block the virus. So there is a way to compress a little bit this timeline, which is what is called drug repurposing or repositioning. So a drug repurposing is based on the idea of taking molecules which are already approved for a specific disease and try to use this drug to treat another disease. So the idea is take whatever is already approved and move it forward and test and hope that something will be able to block, in this case, COVID-19. So COVID-19, you all know that is a disease caused by SARS-CoV-2. And it's really impressive and scary how this virus was able to spread all over the world in such a short amount of time. So yeah, the first alert from WHO was in December 31st, actually. And the declaration of a pandemic was occurring in March 18th. So in less than 2 months and a half, this virus from appearing was declared a pandemic. And up to today, the number of cases raised up to almost 5.6 million worldwide, and the number of deaths is around 350,000 cases. So deaths, sorry. So here is the word, and here is where I'm going to start telling you the story of how we started to look for antivirals able to block COVID-19. So, okay, we are in San Diego, La Jolla, Sanford, Burnham, and Prebys Medical Discovery Institute. And what we do in the Chanda lab is in general, I mean, it's a complex lab, but there is a part of the lab which is focused on drug discovery and especially on antivirals. So we have several collaborations ongoing with another institute not far away from here. So the Calibre Scripps Research, again in La Jolla. And we have several collaborations ongoing for screening chemical compounds able to block viral infections. So of course, as soon as we saw what was happening with this virus, we right away thought about trying a way to apply what we are able to do, so drug discovery against viruses, to find antivirals able to block the virus. So Calibre Scripps developed a library, a chemical collection of several approved drugs and drugs not approved but in advanced stages of trials. I will talk to you later about this. So we directly thought about that collection. It is a collection of around 12,000 chemical compounds, and we knew that they're already advanced, so this was the best way to go for drug repositioning. This is probably the biggest drug repositioning library available worldwide. So the only issue was that at that moment, the virus was still not in the US, luckily not in among the population, but also not available for lab research. So what we did was actually getting in contact with a laboratory in Hong Kong, at the University of Hong Kong, and this is the laboratory of Dr. Yuan, who is actually one of the professors who were mainly involved in the discovery of SARS-CoV-1 back in 2003. And of course, we had the abilities of screening and the library. They had the virus, so why not starting this crazy collaboration and try to figure out if we can actually try to identify drugs able to block the virus. Well, it wasn't that easy, of course, because there were several limitations in the process. But I will talk to you about the difficulties later. First of all, I will give you some more information about the reframe collections. So as I told you, Calibre at Scripps did an amazing job putting together a library of around 12,000 chemical compounds. And these compounds are for 40% FDA approved drugs, so totally safe and approved drugs. And then 60% are investigational new drugs. That means which are drugs not yet approved, but for which there are already clinical trials ongoing. So there are some safety profile data available. So also in this case, these are drugs that can easily be moved directly into patients once validating in animal models. And then there is a 3% of preclinical compounds that require a little bit more investigation for before moving to patients. And yeah, this, as I was mentioning, is the biggest collection of FDA approved and preclinical and clinical drugs. And so it's the best chance of looking for repositioning of chemical compounds right now. So yes, as I was telling you, we had the screening abilities, they had the virus. Well, let's face the difficulties in the process of screening in long distance. So first of all, yes, the library was here, and the library is put in plates, which look like this. Actually, they are this size, so not very big, and there are in each plate 384 wells, so 384 spots, where we can put in each slot one different compound. And this means that up to 384 compounds could potentially be tested on each plate. And of course, this is a massive amount of work. So we have machines that help in this work, which are dispensers. And the first challenge was to ship everything in Hong Kong and be sure that everything was arriving safe. And then the second challenge was to unpack everything and try to explain in distance to people who have never run large content screening, how to screen in such a small format, such a big amount of compounds, right? And this is a part of my job for a while. So this collaboration started so the connection started at the end of January, and we shipped everything in the beginning of February. So for 1.5 months, I was kind of living a double life. I was working in the lab during the day, and at night, because there are 16 hours difference between La Jolla and Hong Kong, I was getting in contact with the laboratory in Hong Kong, and especially with this amazing scientist whose name is Shuo Feng. That was working in the lab, and so we were communicating through a smartphone. And through a smartphone, I could guide them in the whole process of drug screening and large-scale drug screening. So I could teach them how to use the dispenser. I could teach them how to implement the assays, and So then they were running the essays. The essays were run in a BSL-free environment, so I couldn't be physically there, of course, and I couldn't be physically there with the smartphone either. But as soon as they getting out, they were updating me with the data, and I could analyze the data and troubleshoot through the entire process. So this was a real challenge, and I think we are really proud of what we did, because it was, at the beginning, we were not sure that we could have done what we did. And our PI liked to say all the time, well, we're building the plane while we are flying. So we try to do what we can. We hope we will land safe, and we hope we will be able to save people by landing safe. So now I will give you some more details about the screen process. So the idea is that a virus, when it enters a cell, it replicates, right? And by replicated, at some point, the cell gets so loaded on virus that it just explodes. And then, as a consequence, we can say that the virus killed the cell. So the idea is that when you put a virus in contact with cells in culture, so in this case, in our plates, in the long term, the virus will kill the cells. So the cells will become round and then detached, and they will die. But if you're able to find a drug able to block the virus, either by targeting the virus directly or by targeting something in the cell required for the virus, then the cells will survive. And this is exactly what we were looking for. So we were putting in contact these cells with one compound per each well in 384 well plates, and then infecting the cells with the virus, and then measuring how much the virus was killing the cells. And what we were looking for was compounds not killing the cells. So compounds that allowed a rescuing of the viability of the cells. So what happened was that we screened in distance. So Shufeng did the manual work, and we were troubleshooting from here. The 12,000 compounds of the reframe collection, and we identified 300 drugs that were not killing, were blocking the virus from killing the cells. In the meantime, the virus arrived in the US, both among the population, unfortunately, but also available for the laboratories. And SARS-CoV-2 is a biosafety level 3 virus. So I think you all saw around these pictures of people wearing these space suits. And yeah, this is our BSL-3 facility. And what we did in our lab was developing an assay different from the one developed in Hong Kong. So in this case, we look directly at the virus. So we use a technique which is called immunofluorescence, that consists in having an antibody able to recognize a specific protein of the virus, binding to this protein, and then this antibody will be coupled to a fluorescent protein, and the cells in which the virus is present will become fluorescent, so green, while the cells which do not have the virus, like this one, for example, will not be green. And by quantifying the amount of cells green versus the cells not green, we can go and identify the compounds that reduce the amount of green cells per well. So what we did was to take the 300 compounds which were identified through the screen in Hong Kong and retest them with these different assays in our laboratories. And the advantage in this case was that the distance was definitely not the same between us and Caliber. We're just 0.5 miles away. So we could just drove there, pick the plates on the same day if needed, and have as many compounds as we wanted whenever we needed them. And this was a great advantage in shortening the times for the assays and the validations. And what happened was that among the 300 selected drugs in Hong Kong, we validated 30 drugs working at a good level and being safe and working in a dose-dependent manner. And among these 30 validated drugs, we have 6 top candidates that we are currently testing in more relevant models, including Sandra and Snyder's model, organoid model, as well as if they will confirm in vivo, and then the idea is to move them into patients as soon as possible. And so at the beginning was just SBP, Calibre, and University of Hong Kong, but of course there are more and more collaboration ongoing, and I think this is part of the amazing effort that everybody's putting together. The will of collaborate goes beyond any kind of competition in this case, and this is just amazing from a scientific point of view. So we have now collaboration with UCLA, UCSD, Mount Sinai, Purdue University, Kansas State Veterinary Diagnostic Laboratory, and every week we get requests for new collaborations. So these are just the name of people who are involved in the studies at the moment, but as I'm saying, this list is just becoming longer and longer, and I think this is just amazing to see how everybody's willing to collaborate. Thank you.
Thank you so very much, Laura. That was fantastic. What an amazing story of collaboration worldwide. And that's what I love about San Diego. There's a lot of institutions that, first of all, do great work, but second of all, make sure that other people throughout the world have access to the work and collaborate greatly. Thank you to Bobby from the Sanford, Burnham, Prebys Medical Discovery Institute, who made this collaboration and this talk happen. Thank you so much to the Institute for partnering with us. Now we get to the good stuff. We get to the questions. So we already have some questions for you guys. The first one is, how do you create pluripotent cells? Evan or Sandra, who would like to take this?
Yeah, I'll let Evan take this one since he's done them a lot longer than I have.
So about actually 14 years ago, a Japanese investigator named Yamanaka found, to everybody's surprise, particularly to the surprise of developmental biologists, that you could actually take a cell from an adult, a mature cell, skin, blood, hair follicles, and if you treated them with a cocktail of proteins or genes or chemicals that looked like what was present in the embryo, those cells could be pushed back to act as if they were in the embryo, in the earliest stages of the embryo, called the epiblast, where every cell has the potential to become any cell of the body. In fact, the epiblast is the earliest beginning of the human body. We always thought about cells like that as being the human embryonic stem cells, and that, of course, was very controversial starting around 1998. But it was found that you could even take adult cells and make them act like human embryonic stem cells. So the first thing that did was strip away all of the ethical and political debate about using these embryonic stem cells. The other thing that became very appealing, and Sandra touched on this, is that if in fact you take a cell from a patient with a particular disease, or of a particular gender, or of a particular racial background, or a genetic predisposition of any type, and you push it back to the way it was when in the embryo, you can now make cell types and then ultimately organoids that are patient specific. So you can make female organoids, and you can make male organoids, and you can make African-American organoids and Asian organoids. And because these, they can give rise to all the other cells of the body, what we hope is that we may be able to understand why this disease seems to have this amazing disparity, why certain groups seem to suffer from the disease worse than other groups, why males seem to get the disease worse than females. Now, it may be a very simple explanation. It may simply be that the hormones of the female environment are different than those in the male environment. Or it may be that certain backgrounds also have many other diseases that go along with them. But one, we can begin to sort that out because you can make these pluripotent cells from the patient and they have those characteristics. We can sort out, is it something intrinsic to the cell? Is it something in the environment? But even more important than that intellectual question, if we get this right, we can figure out how every patient can benefit from the drugs that Laura and Xin have discovered, in that maybe there is a difference, and all we need to do is change the dosing or change the frequency or add another drug to it so that all patients can benefit from this kind of strategy. And being able to make pluripotent cells from an adult and put them into the organoids that Sandra is making makes that all possible.
Thank you so much, Evan. I appreciate that. The next question, and that kind of ties into what you said, and it might be slightly hard to answer just because there is no one drug yet to treat the disease. The question is, what are some possible side effects of antivirals that are used to block the virus?
I can take it, or I don't know if Xin wants to take it, but
Yeah, you can take it. Yeah.
I can take it. Okay. Well, it depends on the drug, right? The final goal is to have something safe that will not have side effects. What I can talk about the drug, which is the only one currently approved under emergency situation, which is remdesivir. So this is a drug who was in clinical trial phase 3 for the treatment of Ebola. And it was dropped the phase 3 just a few months ago because other drugs, monoclonal antibodies that Sandra mentioned before, work better than this drug. But it's a drug that blocks the replication of the virus, and it's a step that is concerned among several viruses, several RNA viruses. So when COVID-19 started spread around the world, people decided to test. I mean, this drug belongs to Gilead, so Gilead decided to test this drug to see if it was able to block SARS-CoV-2. And we're lucky, it was the case. It blocks the drug pretty well. It blocks the virus pretty well. But the side effect for this drug, for example, is that in the long term, it can cause an accumulation in the liver, so it can show some liver toxicity. Liver toxicity, I think it's one of the main side effects of drugs in general, because it's where drugs accumulate most. So most of them as a side effect has liver disease. That's why I think it's really important not to stop the research just because remdesivir is approved for emergency use. This means, by the way, that it's not totally approved by FDA. This just means that it works well. There is nothing else working as well. And FDA, considering the global pandemic, decided that in this case, we allow people to treat, doctors to treat patients with remdesivir, but just for emergency use. So, of course, in the long term, the idea is to find something safer and not causing liver toxicity. But it's really a case to case. It depends on the drug. And the idea, the final goal is to find the safest drug as possible with no side effects.
Wonderful. Thank you, Laura. The next question is, so I'm just going to read it. I understand that many HIV patients are taking antivirals. Are they less susceptible to COVID-19 because of that?
Xin, do you want to take this?
Okay, so I can take this question. So I think basically for the HIV infected patient, right? Since the patient have a low number of the CD4 T cell, I think this patient would be more susceptible to SARS-CoV-2 infection. I think like the mentioned that because this patient takes some drug, right, anti-HIV drug, actually, we also found that some of anti-HIV drug is also can block SARS-CoV-2 infection. But I mean, it's kind of a balance, right? I mean, the drug can block the infection, but in the patient, due to the weak immune system, right? The virus kind of like more susceptible to. I mean, the virus is can infect the patient easily, right? It's kind of a balance. So I would say that the answer would be no. Yeah. So I think I think the immune system is more important than the anti-drug effect.
That is a good answer. Thank you so much.
I can push that out a little bit in that we use the term antiviral in a broad sense, the same way we use the word antibiotic. But each drug attacks the virus at a different part of its life cycle. The same way every antibiotic is a little bit different and attacks bacteria at different parts of a bacterial's life cycle. And some viruses share some of the same properties, and some viruses share other properties or don't share properties. So it probably is the case that some of the antivirals that attack HIV are attacking aspects of that virus that are very different than the coronavirus. On the other hand, there are some aspects that they share in common. And what we try to do is figure out those aspects that they share in common. And if there's an antiviral drug that hits that particular aspect, for example, replication, then we give it a shot. If it's completely different, we still might give it a shot, but with very low likelihood of it working. So even the drugs we use now, remdesivir, is borrowed from looking at an earlier virus that was quite similar to SARS-2, but not identical to SARS-2. So we're always trying to do that.
Thank you so much. So I'm going to ask the next question. How is this research funded? Always a good question.
After this, we're going to pass the hat. So right now, in all honesty, most of the research that we're doing is unfunded. The pandemic hit us so quickly before most funding agencies had the opportunity to pool all their resources together to be able to fight this. I should say, however, that two organizations have been particularly nimble in making up for lost time and just jumping right in. One is NIH, which has made available some extra funding for this. And right in our own state is CIRM. CIRM, as many of you know, stands for the California Institute of Regenerative Medicine. And even though you would say, well, isn't that the stem cell center? And the answer is yes, but two things. One, they realized from work very similar to what Sandra is doing, that stem cell biologists did have something to contribute to figuring out of all the drugs that Laura and Xin discovered, what may be most appropriate to go into patients, and stem cells could help with that. So they threw money at that. And also they recognized that they are good California citizens. This is an immediate need, and they would jump to the fore. So I would say for us, CIRM has been wonderful in providing funding, and hopefully if everybody re-ups the Institute in November and votes for it, it'll continue sorting out exactly how we can fight this. But the funding right now primarily comes from CIRM and from NIH.
And on our side, yes, we started with totally unfunded. And of course, there was no funding for this project, as Evan was saying. But then, yes, the NIH put money available. So of course, everybody was applying for grants. So a part of our work now finally is funded by NIH. And we also have funding from the Department of the Defense. So we had already some funding for some influenza studies. And so when we asked for extra money for COVID-related studies, they gave us some money. So this is another funding that we have in the lab.
I should emphasize this is very expensive work. So if anybody knows somebody who wants to become a financial hero,
we're more than happy. And I think that philanthropy is also doing a lot right now.
Thank you. Good answers and a good call for support. I like it. The next question is, what should be the course of action by the scientific community if SARS-CoV-2 has multiple strains present?
Did you want to take that, Xin or Laura?
I think that's the goal of having a cocktail of antivirals. This is definitely the final goal. This is what worked, for example, even for chronic viral diseases like HIV or HCV. This is the best way to go to target different strains and to avoid the appearance of resistant viruses. So when you have a cocktail of antivirals targeting different things that can block the virus, it's like attacking a fort from 3 or 4 different sites. So in this case, either one or the other side will be able to block it. So I think that is the way to block it is just keep looking for an efficient cocktail that can allow to block several strains and the appearance of resistance.
And hopefully once we understand
clinically every year as well, because the influenza virus mutates every year. And we have a vaccine that we have to adjust every year for the different mutations. So we're used to viral mutations. And what it actually takes is a collaboration with the world, because we actually look in the Southern Hemisphere when influenza hits that population, what strains they're seeing. And that's how we develop the vaccines against influenza in the Northern Hemisphere when our winter hits. So clinically, we see this all the time, and we just have to adapt based on all the data that we're getting from other countries.
So the other thing I want to add a comment. I think even there are like multiple COVID-2 strain, right? But I think these firewalls probably share the similar replication strategy, right? So in that way, we can find some like a drug that can effectively or like broadly inhibit the viral replication. So we can also go that way, right? Go that way to find the broad anti-viral drug to block the viral infection. Yeah.
Thank you so much. Another question that we have here is, will we need both a vaccine and antiviral drugs to beat COVID-19? Or if one is discovered before the other, will research continue?
So were you going to take that, Sandra? Or?
No, go ahead, please.
So I think that, you know, as Laura indicated, the way to treat these complex diseases is through very complex, multimodal, multidisciplinary approaches. So I think having both is going to be important. There's no guarantee that a vaccine will be 100% protective. It may blunt the severity of the disease, but then you want to knock it out. Similarly, if we have a great drug, that drug may work for a particular strain, but ultimately you would like to anticipate the next season's virus and have protection already with a vaccine, and then perhaps add to it a drug. And we see this all the time. Right now, for example, for flu season, everybody gets a flu vaccine, but you still take Tamiflu if your symptoms have been fairly severe, or you feel like you're too symptomatic. So I would say that ultimately, if you're a physician and you want to treat your patients, you want as many weapons as possible to use. So one area of research will not preclude the others from proceeding.
Thank you so much. All right, we're coming over time. So I'm gonna pick one more question and then I will put my email address in the chat. So if anybody really wanted to get their question answered and we didn't get to it, I apologize. But you can email me the question. I will send it to the speakers. They will send me the answer back and I will email you the answer. I promise. We'll try to get all the question answered. So there's a question here. With the remaining top 6 candidate drugs that could be found to help treat the virus, what is the timeline for seeing if they do in fact work? And if they are found to be effective, what would be the process or timeline for the FDA approval and widespread use?
I think that right now everything is compressed as a time, right? In a normal case, it would take even by drug repurposing up to 1 year. Right now, testing the 6 candidates will take up to a couple of months, because testing them means testing in more relevant models like the organoid system, but also in vivo, and in vivo experiments can take several weeks, because right now there are not so many in vivo models available. And the ones available are pretty complicated, like there are cat models, there are ferret models, and right now there is a Syrian hamster model that seems to work pretty well. But these are experiments that needs to be planned a little bit in advance. These are not just mice, these are bigger animals, and getting those animals in the facilities, which are BSL-free facilities, takes up to 1 to 2 months to test them. In our case, the biggest challenge is having enough compounds, because right now everybody is contacting chemists to have custom synthesis of compounds in big amounts, because when you want to test them in vivo and then eventually move to patients, you need a big amount. So the bottleneck at the moment is the time required for the synthesis, because some of these of the top 6 compounds are pretty complex from a structural point of view. So I don't know, I can give you a detail, like they require, I'm not a chemist, but when they tell me that requires 7 reactions to the synthesis, it sounds complex to me. And, and everyone is giving us quotes for up to 6 weeks just to have enough compound of what we need for the in vivo testing. But yeah, once validated in vivo, I think in a couple of months can be done. Then moving to patients, if the results are positive. We have the collaboration ongoing in Hong Kong. Dr. Yuan has clinical trials ongoing in 6 different Hong Kong hospitals. So as soon as we have in vivo data, he is more than willing to move directly, if there is safety, of course, correlating, to move directly into patients. So we think that if something works, of course, it could take less than 6 months actually to have a positive compound, at least approved for emergency use, like in the case of remdesivir.
I can give you the ideal case scenario. Things never go as ideal, but
no, exactly.
But of the 6 drugs, for example, one of them, or a few of them, have already been in clinical trials in patients, as far as phase 2, and some are even FDA approved. So what that means is, if they've already been in patients, the dosing is already known, the side effects are already known, the biodistribution is already known. So one does not probably need to go back into animals. And for example, if one of those drugs worked in Sandra's lung organoids, we could probably make an argument and go to the FDA and say, you already have a safety package on this drug. Can we have approval to repurpose this, to redesignate it for another phase 1 or phase 1B, 2A clinical trial? So that is conceivably possible. It's been done a little bit messy with hydroxychloroquine. It's been done with remdesivir. And if there was a drug that already has a long track record in humans, and some of Laura's hits do have that, we could probably go to the FDA and at least say, what more do we need to do? And that could be a fast track.
I'm sorry, I'm trying to summarize some of the answers and type them out also, just to make sure that everybody gets it. So
I would have talked slower, sorry.
No, it's perfectly fine. I'm a relatively fast typer, but I apologize to anybody in the audience for any typos or slight scientific incorrections. With that, I think we've taken all the time we have for tonight. I want to thank our speakers one more time. Thank you so very much for donating your time and expertise for tonight's talk. And thank you, Laura, for adding some information into the answer box. I appreciate it. I hope you all enjoyed this event. I hope you will join the Fleet for future events. If you are looking to find where they are, just go to the Fleet website, fleetscience.org, and you can find all the events there. I also urge you to check out the website for the Sanford Burnham Prebys Medical Discovery Institute. I'm sorry. we have a great partnership, but the title is pretty long. I call them SVP. It's much faster.
We do too.
Thank you for SVP. Check them out. They do amazing work. Sign up for the Fleet's newsletter. Get on the newsletter for SVP so you stay on top of all the fantastic science things that are happening in San Diego. And if you enjoyed this talk, I put a little link in the chat where you can find, where you can make a donation if you wish to do so. That helps us to bring more of these science events to you. And with that, I want to wish you all a wonderful evening, and I hope to see you again soon. Thank you, everybody. Thanks, Evan. Thanks, Laura. Thank you. Thank you, Sandra. Thanks, Bobby.
Thank you. Bye.
Bye, everyone. Have a great night.
Bye-bye, you too.
Bye-bye.
Bye-bye.